Well, thank you everybody for coming to our session. I'm excited to kick things off. Thanks to Mark and Victor for inviting me to speak today. So I'll be talking about reliable individual functional networks and their relationship to behavior. I apologize that I didn't upload my slides too far in advance. They're actually online now on my Twitter, but I realized I didn't put my Twitter on here, so it's ESFin Twitter if you want to find the slides and follow along or find them later. And one caveat is that, especially in this audience, I think it's important to say I'm not actually using the word reliable with any formal mathematical or statistical definition, just sort of can we get stable data from a single individual's fMRI scan. So to give you an outline of what I'll be talking about today, give you a little intro as to why we might want to study individual differences in the first place. Then I'll talk a little bit about the factors going into getting good quality brain data to study these types of individual differences. Talk about the importance of grounding these brain differences in behavioral differences and how to think about getting good behavioral data for these types of studies. And then I'll spend a little bit of time on relating brain to behavior, again with the caveat that any of these topics could be entire talks within themselves, but I'll just share with you a little bit of the experience that we've gained over the last few years trying to do this type of work. And finally, a few slides on how to learn more, both here at OHBM this year and also some suggested reading. So why should we care about individual differences? Hopefully you already do, and I don't have to convince you. I think from a basic neuroscience perspective, sort of moving away from group level averaging and being able to map from individual brains to individual behaviors is going to get us a lot more rigorous science. You know, fMRI is never going to be a causal technique, but I think going for the individual level is going to get us a little bit closer to that. And then, of course, there's the consideration of eventual clinical applications for this type of work. So trying to move away from these categorical approaches where we stratify people into groups of patients and controls and just measure mean differences, trying to replace this with a more dimensional approach where we can measure a continuous variable in everybody and kind of plot everyone on the same axis here and get a more nuanced view of the brain behavior relationships that we care about. So if we can get a handle on this, even in the healthy population, it might help us out down the line with psychiatric applications. And a little bit of background as to how I and my colleagues sort of stumbled into this field. So a few years ago now, we were playing around with some of the Human Connectome Project data, which is a nice big data set with lots of individuals scanned under lots of different conditions. So there were resting state scans, there's several cognitive tasks that they had people do. And what we were originally trying to do was, you know, we calculated a functional connectivity matrix for everybody in every state, and we're throwing it into this big clustering algorithm trying to see if we could classify state. And instead of getting state, we kept on getting out subject. So in other words, we kept noticing that the same subject doing two different things tends to look more similar. You always tend to look most similar to yourself, regardless of what you're doing. And we sort of formalized that into these identification experiments where we noticed that if we just try and match pairs of connectivity matrices calculated from different scan conditions, we could do this with a very high degree of accuracy. So just to orient you to this matrix here, each cell is the accuracy of the identification rate, again trying to match the same subject across different scans. So REST 1 to REST 2 was a 93% accuracy, dropped off a little bit between REST and a task or between two different tasks, but this was an original sample of 126 people, so chance here is less than 1%. We're doing this well above chance. So the main point here is you sort of always look most like yourself regardless of what you're doing, and this was encouraging to those of us that are hoping to study individual differences. We went to great lengths in this paper to try to rule out a lot of the confounds that maybe might be occurring to some of you. I won't go into that since it's mostly published at this point. But I think the even cooler part of this was that we noticed that not only do individuals have this unique, reliable signature of functional connectivity, but features of this could be used to actually predict people's levels of fluid intelligence. So individual differences in the brain were related in a meaningful way to individual differences in behavior. So this was sort of the groundwork for how we got into this and what we found exciting about this. But this original study brought up a lot of questions, both on our end and other people wondering about the factors that go into this and sort of what you need to do to get these types of results. So moving into the next section about brain data. One of the first questions people started to ask was, do you need HCP quality data to get this result? And the answer, I think, is not really. Okay, so higher quality data is always going to be better. I'm certainly not going to stand up here and argue the opposite, but it turns out that this identification result is fairly robust, even at more standard spatial and temporal resolutions. So Aaron et al. Had a nice paper last year in human brain mapping where they looked at identification in some publicly available data sets, which with much more standard voxel size and TR. Here on the y-axis, actually lower is better, so they're using kind of a rank sum identification algorithm, and on the x-axis is acquisition time. So even with just about 10 minutes worth of data on people, you can get this fairly stable identification. And another interesting result, this is from Jason Drusgal down at UVA, sort of investigating the effect of extreme temporal downsampling on this result. So here they're artificially manipulating the TR by just taking volumes at certain time intervals. And he's found that you can get up to, even with a 10 or 12 second TR, you can actually get a pretty stable result. So it seems that you don't necessarily need this super high quality data to observe this. One thing that does seem to make a difference that Aaron et al. Found, and we've also found in our data, is the number of nodes that you use. So more nodes is going to give you a higher identification rate. They, in that paper, noted that the actual method of how you parcelate the brain didn't seem to matter too much. And then, of course, one cautionary note here is using extremely high resolutions, you might be opening yourself up to more effects of anatomical differences across subjects or registration errors. We've looked into that a fair amount. If people have questions, I'm happy to talk about it afterwards. But just on sort of a practical level, I think we and others have sort of started to converge on parcellations in the 200 to 300 node range, giving us fairly stable results. But it's just something to think about if you are interested in doing this type of stuff. What about amount of data? So this is one thing that does matter a lot. This has been shown a number of times now in a number of different ways. Scan duration does matter a whole lot, so longer acquisitions are always going to be better. This is true theoretically, practically, any way you slice it. This is an older result now from Rasmus Byrne showing within single subjects, interclass correlation coefficient increases as a function of your scan time. So that's a within subject analysis. We've also seen this with the identification experiments, unsurprisingly. So the more data you put into your connectivity matrix, the better able you are to identify or to separate subjects from one another. But one sort of interesting nuance here is that a higher sampling rate, in other words, a shorter TR, can't make up for a shorter scan duration. So this has also been shown a few times now. This is another result from that Aaron paper. I know this is a little small, but that data set with the red line is the data set with the shortest TR. That's a TR of about 0.6. And it actually takes the longest number of data points to kind of converge on the best identification. So this is perhaps unsurprising given our understanding of hemodynamics and just the general advantages of scanning people for longer, but that's one thing to note. And here's another result from Shaw et al. Where they're again, artificially sort of chopping out amounts of data, but depending on how you do it, it can have a big effect. So in the red line up there, they're just sort of randomly selecting TRs across the whole scan, and you can see that the interclass correlation coefficient stays pretty high. And in contrast, using the same amount of data, but getting it by just truncating or chopping off contiguous volumes at the end of the scan, you see a much larger drop in ICC. So you can't, you can't make up for less scan time with a shorter TR. In fact, you may actually be hurting yourself, just on a practical level. So, okay, so you have your people in the scanner. You're going to keep them in there as long as you or they can stand it. What should you have them doing in there? So one of the things that this identification result sort of brought up was, if you always kind of look most like yourself, regardless of what you're doing, does it even matter what you're doing while you're scanning you? And I think, yes, it does still matter a lot. So resting state has become the default condition for functional connectivity analyses, and individual differences in functional connectivity are no exception to that. But I know a lot of other people, I think, are starting to come around to this idea that we shouldn't be so quick to throw out tasks. Tasks may actually increase your signal to noise in a lot of ways, especially for individual differences. So bringing back this original result here, one thing we actually didn't do a great job of in this paper was controlling for amount of data per condition. So in the HCP data set, the rest acquisitions are by far the longest. They're about half an hour each. And the next longest task, I think, is the working memory, which is only about 10 minutes, and then the tasks decrease from there. So if we look at this in a way that we control a little bit better for amount of data per condition, this is an expanded HCP set. This is, I think, this is the 900 subject release. And here we're looking at, these are correlations within subjects. This is the average correlation within a single subject. Do I have a pointer? Yeah, between the different scans. So, for example, REST 1 to the working memory scan, what's the average correlation within a subject of their functional connectivity profile? And this upper triangle is using all of the available data per condition, and the lower triangle is truncating everything to be the same length. And you can see that if we use all the available data, these two REST scans have this sort of privileged, amazingly high correlation between them. But if we reduce that down to the level of the rest of the tasks, that privilege status kind of goes away. They're still fairly high, but not nearly as high as that. And you'll notice that if we control for amount of data, there's actually pairs of tasks that are even more similar to one another than the two REST scans. So these are two different tasks achieving higher similarity within the same subject than the two REST scans, which in theory is the same condition. But as we know, it's very difficult to constrain and to know exactly what subjects are doing during REST. So this is just a hint that for a set scan duration, tasks may actually be more reliable than REST in the individual. And another way of thinking about this is that tasks might converge faster on a subject's true connectivity profile. So moving along with this theme, is REST best? Is REST the best condition? And I'm going to say probably not. We notice, so that the previous slide was all sort of within subject. If we extend these identification results to this larger HCP data set, we see similar things. I apologize if this is kind of small, but the point here is that the rest-to-rest accuracy is around 70%, which again is pretty good considering the chance is less than 0.1%, but there's certain task pairs that get even better. So the relational to the working memory, for example, is at 83%, even though these are two different tasks acquired on 2 different days. And if you have the time or the inclination, you might consider using a combination of rest and tasks. So this is a result from the original paper where we found that if we showed our model a rest and a task scan from the same subject and then showed it a different task scan, it was more accurate at identification than using just the rest or the task matrix on its own. So if you can do both, that's great, but you might want to consider using tasks instead of rest. They might get you more bang for your buck. One thing that I've become really interested in lately is naturalistic tasks. So if this will play, yeah, so these are things where subjects are listening to stories or watching movies in the scanner. This is just a cool example from Tammy Vanderwall at Yale. She developed this movie called Inscapes. It was originally developed to try to increase compliance in kids who are not very tolerant of just doing resting state in the scanner for long periods of time. So she had them watching this video. And it's pretty cool. Unfortunately, I don't have time to show you the whole thing. It's pretty mesmerizing, but you can watch it at that website there. And we had been collaborating with Tammy, and we were interested in sort of what does a naturalistic task do to the individual functional connectivity signature. So you might think that by imposing the same rich stimulus on everybody, we're going to be evoking patterns of activity, and therefore connectivity, they're going to be really similar across subjects, and maybe we're going to wipe out our individual differences. But that's not, in fact, what we find. So Tammy had a test-retest data set with people resting, watching this Inscapes movie, and also watching this fast-paced social movie. It was a clip from Ocean's 11. And we actually found that identification was still really, really high across these 3 tasks. So if you look down the diagonal here, these would be between pairs of the same conditions, so REST 1 to REST 2, or Inscapes 1 to Inscapes 2. And those are really high, but the off-diagonal elements are also really high. So you still look quite similar to yourself, regardless of whether you're resting or watching one of these videos. And I think that's a cool result in favor of naturalistic tasks. And if you're still not convinced, tasks also have purely practical advantages. So they increase subject compliance mainly by decreasing head motion, especially in certain populations. So here's a nice paper that just came out showing see this plot here. These are different populations. So here all the way on the left is controls, and then there's 3 different psychiatric populations. So the patients are always going to move more. That's hard to avoid, but This first bar here in red is the resting state scan, and the rest of the bars are different tasks. So pretty much everyone moved more at rest than they did during the tasks, and that's echoed down here. The first and the last scans were the rest scans, and they had higher motion than the middle two task scans. Tammy Vanderwall has also shown this in her data in both children and adults. So people move more when they're resting versus watching one of these sort of fun videos. So you might consider using a task just for purely those reasons. But moving right along, since I'm already running low on time, so what do we want to think about in terms of behavioral data? What are the considerations that go into choosing an interesting behavior? So the first thing you want to think about, at least for a cross-subject study, is it stable? So is it fairly similar if I measure you today or tomorrow or next month? Is it going to be more or less stable? So thinking about things like trait versus state. So state variables might be quite interesting, but they're probably better suited to a within subject analysis. This was a cool result just came out from Rick Betzel on the Russ Poldrack myconnectome data, where he found functional connectivity correlates of things like surprise and positivity. So these are traits, maybe Russ will disagree with me, but fairly variable across different days. So again, very interesting to study from a within subject perspective, maybe less meaningful from a cross subject perspective. They also found that fatigue strongly mediated this positivity relationship here. So again, that's not necessarily a trait, might not be best suited for in a cross subject analysis. The next question you want to ask is, does it show a good distribution in your population? And it's really important, I think, to just look at the distributions. One sort of cautionary example from the HCP data, again, is if you look at the distribution of performance on the emotion task, there's a strong ceiling effect such that this task seemed to be fairly easy. Most people are performing around 90 or 100%. So, maybe it seems obvious, but it might not be that interesting to look at individual differences in a measure that doesn't show a lot of differences. In contrast, something like the relational matching tasks seemed to be a lot more challenging, and so they got a much wider spread of behavior on that task. That might be a better target for in a cross-subject study. And then finally, just a quick sanity check. Is it something that you actually expect to be reflected in brain function? Do you have a theoretical motivation to believe that this is something that could be predicted from intrinsic or even task-evoked connectivity? And I won't give specific examples, but just ground yourself and ask yourself if this is something stable and trait-like enough that you might expect to find neural correlates of it. Okay, again, on a practical level, mitigating confounds, and again, each of these slides could be a whole talk in and of itself. Hopefully this is obvious by now, but many behaviors and phenotypes are highly correlated with head motion, as has been shown over and over. So a recent paper by Siegel et al., looking again in the HCP data set, found fairly strong but very systematic correlations between head motion and several measures. So people that did better on a lot of the cognitive tasks tended to move less in the scanner. Perhaps unsurprisingly, people that had a higher BMI, higher scores on DSM measures, etc., tended to move more. These are pretty pervasive. Of course, you have age of effects such that young kids tend to move more and older adults tend to move more. On the aging side, this may even be related somewhat to vascular health. This is another result that just came out a couple weeks ago by Geerligs et al. In human brain mapping. So these are things to be aware of. Hopefully most of us are aware of these by now. What can you do about it? Well, you check for the correlation in your sample, so you can't control for it if you don't check for it. You can consider excluding particularly high motion subjects if you have a few outliers that are really driving your correlation. You want to be careful subsampling too much where you can introduce bias there, but if it's just driven by a few people, you might want to at least rerun your analysis in a sample where the correlation is lower. Choose appropriate pre-processing techniques. This, again, everyone's going to have their own opinion on this. I think it's just really important to know what you're doing in pre-processing, and this was another paper that I just saw. Again, there's been countless publications on pre-processing and mitigating motion effects, but this one just came out. I think it's particularly striking. So all the way over here on the left is if you just do your standard 6-parameter motion regression, most of the brain ends up being still correlated with head motion. And as you move down these pipelines and get a little bit more sophisticated, you can get down to slightly more palatable numbers of edges that seem to be correlated with motion. So I won't give you any magic bullets on this one, but think about it and know what you're doing to your data. And then, of course, you can always use motion as an explicit covariate in your behavioral prediction analyses and things like that. Okay, so how are we going to relate the brain data to the behavioral data? Cross-validation is the best way to ensure that what you're seeing is rigorous. So what this means, for those of you that don't know, is that if you find a brain behavior relationship in one sample, you want to see if it holds in another sample. You can do this in a leave one subject out sense. It may not be the most ideal, but it's certainly better than not trying at all. So even if you have a small-ish data set, you can try this. Bigger numbers are always better, but I'm going to go through this quickly. I think most people know what leave one subject out is, but you can iteratively leave a subject out, build your model on the rest of the data set, and then predict for that left out subject and move on through. You can do this in a k-fold way where you're leaving out more than one person. But the idea here is that if you see a correlation between your predicted behavior and your observed behavior, that's a clue that maybe what you're seeing is going to stand up to validation in an external set. And we have a paper out in Nature Protocols that describes just a really simple, this is extremely easy to implement, doesn't require a lot of fancy machine learning. Those techniques may get you better results, but this is just, if you're new to this and you're looking for something simple to implement, check out that paper. An even better way of doing cross-validation is cross-data set. So Monica Rosenberg in our group has some really nice results with predicting sustained attention, where she trained a model to predict attentional performance in a sample of 25 adults scanned at Yale, and then showed that the exact same model could predict symptoms of ADHD in the opposite direction, of course. So better sustained attention implies lower ADHD symptom scores in an entirely independently collected data set from Peking University. And there's a lot of cool publicly available data out there. So if you have hypotheses or things that you've observed in your own data and you want to try to test them in an external set, this could be a really nice way of doing that. The final point I want to make is the best brain state for predicting behavior may actually depend on the behavior that you're interested in. So one final interesting observation that we made in the HCP data set was that if you feed your model connectivity matrices that are acquired during different conditions, there's a decent amount of variability in how well you can predict behavior. So here are the different matrices from the different HCP conditions, and here we're trying to predict out-of-scanner behavior. So this is fluid intelligence. We had the vocabulary measure and the list sorting task, which is a test of working memory. And we found that the rest scans were actually some of the worst predictors. So these are still technically significant in a 10-fold cross-validation, but the better results came from using the task scans themselves. And again, these are behaviors that are not in the scanner. So these are outer-scanner measures, but it seems like if you are sort of giving your subjects a cognitive challenge, you're drawing out some variation that's going to be relevant to these cognitive abilities. And so you might consider tailoring your scan condition to your behavior of interest. I like to call this a stress test. And I'll be talking a lot more about this at my poster and also at our symposium on Wednesday morning if you want to learn more. So just to hopefully wrap up now, a few take-home points on the brain data side. Data quality is helpful. It's probably not critical. So if you have some older data sets lying around the lab and you want to try these analyses on them, just if you don't have the HCP sequences, don't let that stop you. Longer scans are certainly better. You might consider using tasks instead of or in addition to rest. They improve compliance as well as potentially increase your sensitivity for finding correlates of behavior. On the behavioral side, you want to choose what I'm calling an interesting behavior with a good distribution in your population. And of course, as always, you want to consider potential confounds and take as many steps as you can to mitigate them. In relating brain to behavior, cross-validate whenever possible. And again, consider tailoring your scan condition to your behavior of interest. Okay, so that was sort of a whirlwind tour. If you're interested in learning more, here are just a few of the open data sets with brain and behavior. Again, many of you are probably familiar with a lot of these, but don't forget that they have the behavior as well on a lot of these. And so that's a really cool resource. You can use them on their own or in combination with data sets that you have in your lab to either generate or test hypotheses. Here are some reviews. Again, a lot of ink has been spilled on this topic. These are just three that helped me out, plus a shameless plug for my own review if you're interested in. And these will point you to a lot of the primary literature that I talked about today. These are review papers. And finally, if you'd like to learn more here at OHBM this year, we have a symposium going on Wednesday morning called Relating Connectivity to Inter- and Intra-Individual Differences in Attention and Cognition. There's a similar symposium Tuesday morning that looks really great, and a bunch of us have posters here, and we would love to talk to you more about this. So thanks so much for your attention.